Welcome students. Today we are going to see the study of anti-ulcer activity using Pylorus ligation method. So this is one of the experiment of pharmacology 3. So let us start with the principle of this experiment. So peptic ulcer is one of the relevant prevalent gastrointestinal disorder, and today we are going to study it with the using the Pylorus ligation rat model and or mice model. So here we can see that this method was first demonstrated by the Shay in 1945. And what is the principle behind it? We can see this ligation, or that tying the pylorus of the stomach, that will increases the gastric acid accumulation in the stomach, that leads to the acute gastric ulcers. So this procedure used to screen the drugs for their anti-secretory and anti-ulcer activity. So what is the principle? That the free gastric HCl which is accumulated in the stomach will cause the ulceration in the stomach, and this ulceration reduced or prevented or inhibited by the various drugs that we are going to screen. And today we are going to demonstrate the anti-ulcer activity of the omeprazole drug, that is one of the proton pump inhibitor drug in the category of peptic ulcers. So let us start with the experiment here. So what we have to do for the carrying out this experiment, we have to use the rats which are fasted for 24 hours before the pyloric ligation. We have to divide the rats into two groups. One is the control, that is vehicle treated group, and another is the drug treated, that is omeprazole treated group. Then after that we have to administer the drugs, and before 2 days we are administering the dose, and 30 minutes prior to the ligation. Then first thing we have to perform the survival surgery by making the rats anesthesia, giving the rats ether anesthesia, and we are making the pyloric ligation procedure by making the surgery. Then these rats are placed into the separate cages, allowed to recover, and after 19 hours of pyloric ligation we will sacrifice the animals by decapitation method and particular ethical procedure. Then taking the abdomen, opening the abdomen by surgery and taking the stomach out of the body. Then the contents of the stomach will be collected, and the stomach will be cut open from the greater curvature, and the ulcer production or the ulcers observed under the magnification of 10x, that is the microscope. So this is the procedure and this is the instruction given. So according to this we have to perform the experiment. So here we are using the this virtual practice by Health Education Bureau. So let us administer the drugs for the rats or mice. By this procedure we have to administer the drugs to the rats specifically. So here you can see this video is showing how the drugs are administered and how the pyloric ligation survival surgery is done. In this video we are performing the pyloric ligation. Now pylorus is the end of the stomach, the last part of the stomach, where this is the sphincter, and sphincter means wall. This is get tied with our thread, and after tying with the thread, we are allowing the stomach to accumulate the HCl. This HCl may cause the ulcers in the stomach, and that ulcers we are going to check after 19 hours of this surgery. So we are taking the stomach here out of this. You can see here we are taking the stomach out of this abdomen and tying it with the thread. You can see this thread is tied here, and we can again make that stomach inside, put it inside the abdomen and making the surgery done, so that the animal will survive for next 2 days. So after performing this surgery, we are separating these animals in the separate cages to maintain their recovery. We have separated the animals, vehicle treated and drug treated. So after we have to check out the observations. So for checking of the observation, we have to open the animal abdomen. We have to sacrifice the animal by decapitation method, and we are taking out the stomach. And after taking out the stomach, we are opening it by greater curvature. So here you can see we are opening it by the greater curvature. And now we are observing the inner side, epithelial side of the stomach, and observing for the production of the ulcers, and we have to score that ulcers. So how we can score? The mouse one will show these kinds of ulcers as shown in the diagram here. So we can put 2 ulcers in the rat one. Second, again the same observation we can see in the mouse 2. 3 ulcers count. In the mouse 3 we have seen 4 ulcers count. The same procedure for the drug treated group. If we have saw here the less ulcers, we can score them 1. Some in the next rat or mouse we have seen no ulcer production. In the third rat or mouse we have seen again there is no ulcer production due to the drug treatment. So by taking these observations, we can make this kind of observation table. Here in this observation table you can see the vehicle treated group, that is the control group, shows the more number of ulcers production, that is average 3 ulcers are produced in the rats or mice, while the second group, omeprazole group treated, it shows the less or no ulcer production. So by these observations we can conclude that the higher numbers of ulcers count in the vehicle treated group than the drug treated, and this reflects the anti-ulcer activity of the drug, that is omeprazole. So omeprazole protects or inhibits the production of ulcers, and this is we have demonstrated that the anti-ulcer activity of omeprazole is confirmed. So by this experiment we can see that anti-ulcer activity, omeprazole shows the anti-ulcer activity. That is the today's experiment. So after getting this experiment, I like to request you to share this experiment or this video in your friends group, so that they will get this benefit of this video and the experiment. Thank you.